What's that? Oh, no. Okay. Right. Okay. Okay, folks, let's go ahead and get started. So the first chapter in the Maclaury book talks about different types of inheritance patterns. So I have here some pedigrees, and I hope we can use these to try and figure out different types of inheritance patterns of these three different traits. The next chapter is about diagnostic processes to try to test someone for a genetic condition. And then the next chapter looks at prenatal diagnosis and then genetic counseling and the like. And then it gets into some more of the different types of actual genetic traits that you're most likely to find. Very good. Let's go ahead and get started with a word of prayer. Lord, thank you for this cold day. Thank you for every student here. Watch over us, please. And we are very thankful that you've kept most of us safe and well so far. Please continue to do so. In Jesus' name, amen. Okay. When you look at this trait, you notice that we have two parents here. One seems to have the trait, the other doesn't. Let's just put some kind of flesh on the bones here. Let's say this individual here, let's just say it's recessive. And maybe this person is either this or say is this. So we don't really know here. We're just trying to see if this works. All right, so then we have these three individuals. Two of them are affected. And then we see that, so this individual here, this would be person number 2. This would be person number 3. And this would be person number 5 over here. And to make this clear, let's actually make this. Now, these two people are affected. That means if this is going to work, this individual has to be heterozygous, right? There's just no other way these two people can be affected. This person is homozygous for the recessive trait, for the wild type trait. All three of these individuals should be heterozygous and they should not be affected. So all three of these individuals, were this to be the case, would be this. And none of them would be affected. So this person, if this is going to work, has to be this. All right, so if this person is this, that means your progeny have to be either this genotype or this. So this person's not affected. That means this person would have to be this, right? Yeah. And therefore, this person is probably this, and this person is probably this. Yeah. Good. Okay. So then we have this person with number 4, and then this person with number 6. Yeah. Okay. And then we get 2 folks here. We get that's the, and then we have the 3rd generation here. Now this individual then goes on Okay, here, and that's individual number 1 right here. And we have this daughter here, this daughter here. Well, this could work if this person were this, right? That means half of them would be this, and half of them would be this. So this person could be either this, and this person might be this. That could work, all right? Now, if this person is this, then none of their progeny should be affected, but they're all affected. So at the very least, then, this person would have to be almost heterozygous, right? So if that's the case, then we would expect half of them to be affected, and it turns out they have 2 kids. All of them are affected. So that means this one would be this, and this one would be this. That's 3, and that's 4. And then over here, we'd have to have essentially the same situation, because number 5 over here is affected. So that means this person has to be this, which means this person also would have to be that, right? And then that means this person here, number 6, that person's not affected. Sorry. This person would have to be this. Now, for this to work, that means this allele has to be relatively common in the population. What if that's not the case? Those are the 2 individuals here. That's 1, and that's 2 of the 3rd generation here.
Are they both like capital A, capital A?
We don't know. We don't know. They're either if because progeny essentially from that mating half of them will be this genotype, half of them will be this genotype. So they're not affected, so they're either this one or this one. If the section, so this person here, for this to work, this person has to be heterozygous, has to be. So these individuals are either this genotype or this genotype, we don't know which. All right, so the assumption then we have to make, so with this mating here, this person has to have the affected allele. So does this one. What if that's not the case? Also, you have to assume that this grandparent here has the affected allele. Now, that might be the case. You might have an allele that's pretty common. What if that's not the case? Then this doesn't work. What works better then is this trait is dominant. And so instead, have this sort of situation. Like that. So then this individual has to be a heterozygote. So does this individual. And then this person is unaffected. These people are unaffected. So all of their progeny then will be unaffected. That works quite well. These individuals here, half of them will have the dominant allele, half of them will not. So this one is probably that, this one's probably that. And then over here, we have half of them being affected and then half of them not. That works much better. So this pedigree then is best explained, most parsimoniously explained by a dominant trait. So if you see this type of pedigree, you can work it out with the recessive inheritance pattern, but you have to make a big assumption. That assumption meaning that recessive allele is pretty common in the population. Now you might say, well, they're from a really small town where people tend to marry each other and no one leaves to go to college or something like that, or They go to the local junior college and they all stay there or something like that. We're all familiar perhaps with small towns like that. Heck, Spring Harbor might be a small town like that, but we won't get into that right now. But anyway, in many cases, genetic diseases, genetic conditions that you're going to see, they're not that common. So that is a big assumption that you can't make. So this is best explained by dominant inheritance. This, in fact, this pedigree is of a dominantly inherited trait, or genetic disease or whatever. Okay, let's look at this one. What do you see about this one? Not common. Well, you have two parents up there that don't have the trait. They don't have the phenotype. And all of a sudden, pop, there comes one of their progeny who has it. And oh, look over here. We have these two progeny who don't have the trait, but then pop, out comes one of their progeny who has it. And then, oh, look over here, pop. There we go. That is really common when you have a recessive trait, because you have two individuals who are heterozygous. And if they're heterozygous, there's a chance that 1/4 of their progeny will have that trait. If you don't believe me, just run the Punnett square. Let's go ahead and run the Punnett square. Come on, do it with me. Get your stuff out, do it with me. So 1/4 of the progeny are affected, right? 3/4 of the progeny are unaffected, but half of them will be carriers of the recessive trait. 1/4 of them will not be carriers of the recessive trait. Okay, I did that kind of fast. Want me to explain that again? Yeah, so when you do a Punnett square, you take the alleles that the parents have and you just separate them. The convention is usually it goes mom pop usually, but it really doesn't matter. You can go pop mom, or I just tend to do mom pop. That's just me. I try to give my wife the position of honor. Anyway, so you have the two alleles like here. Remember, you're going to separate gametes during meiosis. Those gametes will then come together in a random pattern according to Mendel's law of random assortment. So you just do the matings, big A, big A, big A, little a, big A, little a, little a, little a. So there's 4 possibilities. Out of those 4 possibilities, only one of them will give you a homozygous recessive condition. How many progeny do these folks have? 3. 1 out of those 3 has the trait, even though the parents didn't. Then this couple here, their single daughter did not have the trait, but now her progeny, here comes the trait right here that says that this person is heterozygous. So this is an example of a recessive trait where you see the trait essentially skipping generations. That's really common with the recessive trait. So this is a pedigree for a autosomal recessive trait. So that one was a dominant autosomal trait. This one is a recessive autosomal trait. And I think you can see qualitatively there are differences between the two. The other one, every generation, you had the trait. Someone had the trait. Not so here, you have it skipping generations. Okay, see the difference? Now let's look at another one, and you're going to see something kind of interesting. Come on. Stop you. Okay. Why did it do that? Okay. Okay, yeah. There. Oh, okay, that's the one I want. Okay. There. Bang. Okay. All right, now what do you see here? It's in every generation, but look, the female has it, then the males all have it. The female is a carrier, now only the male has it. They both have it, everybody has it. That's an example of a sex-linked trait. It's on the X chromosome. Males have only one X chromosome. So if they get the X chromosome with that particular allele that gives you that trait, only the males will inherit that trait. The females will not. Because the females get an X chromosome from the mother and an X chromosome from the father. So if the daughters receive the X chromosome with that recessive allele on it, they won't show it because they have a dominant allele that they receive from their father. The males, on the other hand, just have the Y chromosome. So they don't have a second allele of that X chromosomal locus. So only the males are going to show the trait. The female here in this case is a carrier for the trait. Only the male, however, will show it. The females will not. Well, she's showing it because both the males and the females have the trait. That means all the X chromosomes have that allele. So regardless of which X chromosome she gets, the two she gets both have that allele. So there are several different genetic diseases that you will probably see in your careers. Hemophilia A and hemophilia B are both X-linked traits, as is something like Fragile X syndrome. Chronic granulomatous disease, muscular dystrophy, those are all X-linked traits. So if you do any family medicine or pediatrics, those are traits you will most likely see. And this is the type of inheritance pattern that you'll see, where you go from female to male and so on and so forth. All right, so now go back to our slide. Now this talks about some of the different patterns. So with the autosomal patterns, this is essentially the inheritance pattern you'll see. Now, if we did a Punnett square, say, for an X-linked pattern, we'll do that here in just a bit. Again, with the dominant inheritance patterns, you can have a 50% chance of inheriting the affected allele. With a recessive pattern, you will have a 25% chance of acquiring the affected allele. If you have so-called consanguineous matings, that is, you have matings between first cousins or something like that, the chances of seeing recessive traits goes way up. And that's one of the reasons why consanguineous matings will tend to usually greatly reduce fertility, because typically the babies will die very, very early during development, because it turns out we have recessively lethal mutations kicking around in all of our genomes. And if you happen to marry someone to whom you're closely related, which happens in some communities unintentionally, you start to see a higher trend towards certain types of rare genetic diseases. When I was at the University of Pennsylvania, there's the Child's Hospital of Pennsylvania, and then the Children's Hospital of the University of Pennsylvania. They're respectively called CHOP and HUP. And CHOP and HUP have this long-going research program with families that have these rare genetic diseases. And in Pennsylvania, you got this ready-made group where you have a lot of marriages between people who turn out to be closely related, and that's the Amish, because you have a closed community. And so eventually, if you marry someone within that community, it's going to be someone where usually you're going to share some family history along that line. And it was very common to see Amish families essentially coming into the hospital with their very distinctive dress and their children usually had some sort of genetic disease. I distinctly remember we lived on 44th and Pine, and so I was in walking distance from where I worked, and I would usually come out, and the parking lot was usually over here. And the Amish, these Amish, at least they don't drive, so the university gave them some sort of transport, and they came out, and I distinctly saw this little girl. She was dressed in Amish dress. Her dad was carrying her. She had 6 fingers on both hands, and I remember her going like this. And I remember going, 6, whoa. And she also had some sort of facial condition. It was difficult to describe. Her face just didn't quite look normal. And that was a family that was participating in that sort of study. And if you work in communities that have been very, very tightly knit for quite a while, you'll find more of this than you think you will. Now, for X-linked inheritance patterns, mothers will not pass it to their daughters unless the father is also affected too. A good example of an X-linked trait that's a bit more benign than, say, hemophilia or something like that is color blindness. So there's 2 types of color blindness. One is X-linked, the other is autosomally linked. So let's just take an X-linked trait, for example. So let's say we have this individual, say, is red-green colorblind. That allele is on that chromosome. So this one here, the allele is normal. Let's say she has kids with a chap who is not colorblind. Okay, so this individual here, this daughter will not be colorblind, but she'll be a carrier for the colorblind allele. This individual here is going to be colorblind. This individual will be colorblind because there is no X chromosome with the wild type allele to oppose this colorblindness. So this chap is colorblind. This lady here is not a carrier. This individual here, that individual is a male and they are not colorblind. So you can see in this case, all the color blindness is found in the male. Females will not be colorblind. The mother will not pass the color blindness to her daughters. That is a feature of X-linked patterns. There are a few dominant X-linked genetic diseases. They're really rare. Unless you work at a university hospital that specializes in things like that, you probably aren't going to see any. So we'll just dispense with that because the clear majority of your X-linked traits, your X-linked genetic diseases are recessive. There's only a few that are dominant.
So the X is the positive is what makes it.
If you have this allele, color blindness allele with the Y chromosome, that person is colorblind. For the female to be colorblind, she'd have to inherit 2 of these alleles, and she can't because there aren't 2 of them to inherit. And that's usually the case. Now, if you get a carrier who has kids with a colorblind dude, yeah, then you can have a daughter who's colorblind. So if Anne was colorblind, carried a colorblind allele, and then one of Aaron's kids his daughter Tessa, half of her daughters would be colorblind. But fortunately, Anne sees color beautifully and is not a carrier for colorblindness, because Aaron is really colorblind. When he got here, we used to play some dirty tricks on him, but we don't anymore. All right, and that just tells you the story. Excellent, yeah. Okay, mitochondrial inheritance patterns become important if the mitochondrial genome experiences a profound mutation. These profound mutations are usually deletions of certain genes. These particular mitochondrial inheritance patterns are inherited through the mother, simply because the egg is a far bigger cell than the sperm. The embryo will get a few mitochondria from the sperm, just a few. The embryo gets far more of its mitochondrial genomes from the mother. So these genetic diseases are passed from the mother to the offspring, not from the father through the offspring. So they show so-called maternal inheritance patterns. Maternal inheritance patterns. So they're only inherited through the mother. And the Human Genome Project, of course, has provided the means to try and find a way essentially to customize somebody's care. I think the hope of this was a bit oversold. It has turned out, I think, in some cases to be quite useful. In other cases, this idea that you could essentially take some cells from inside someone's mouth and essentially get a readout of what drugs will do the best job to treat this or that or the other thing, it hasn't really come to that. So that hope was a bit oversold, shall we say. All right, so quick summary. Germinal mutations occur in the germline. They occur in the cells that make the gametes. They're passed on to the offspring. Somatic mutations are not passed on to the offspring. Somatic mutations occur in the body. They occur in the body. Rates of non-disjunction go up, and as maternal age goes up. As paternal age goes up, mutations can accumulate in the germ cells as well. And for a while, your textbook said very little about the effects of paternal age. Now there's an appreciation that paternal age actually can contribute to the genetic load of the infant as well. And then these are inherited in Mendelian patterns that we talked about. The study of the genes as a whole within the genome, that's what we mean by genomics. Genomics. All right. Are there any questions about Mendelian inheritance patterns, reading pedigrees, that sort of thing?
I guess for me, like when it comes to the pedigree, how can you just like look at it and decide whether something's like, I don't know, like capital A or how do you tell?
You try it out essentially, just like what I did with that pedigree. You try it out. And what you assign them as is usually arbitrary. You're just using symbols as placeholders to help you think about it. They're used, the term that we use is their heuristics. That's okay. You just assign it as something and you see what inheritance pattern works. And you use the inheritance pattern with which you have to use the fewest assumptions. The fewest assumptions, so you use the most parsimonious explanation. That's what you use, and that's usually what works. 19 times out of 20. Occasionally you get fooled, and you're going to get pedigrees usually through medical histories. In Great Britain, they will actually give people little diaries to take home and they have you fill out these questions, and they have you in the with the diary. You write in the diary because things may come to you as you either look things up or as you think about things or as you call people and get answers. You write things down in the diary, and then you come back to the doctor and you present the doctor the diary, and the physician or the genetic counselor or the nurse sometimes will look at the diary and fill out the information and try to make a pedigree with some more information and what have you. So pedigrees, they require a bit of detective work sometimes. All right, so a bit more about pedigrees. Now, if you have in a pedigree a box that is tipped on its side like that, that means this person had a baby, have no idea what the gender of the baby was, no idea if the baby was male or female. So that if you just don't know, maybe the baby died really early and nobody thought to write down, you know, boy or girl or something like that. So in the pedigree, you'll indicate that with a rhombus, essentially like that. And we talked about essentially, if it's filled, that means they have the trait. If it's not filled, they don't have the trait. If you know someone is heterozygous, you take the circle or the box and you half fill it. That means they're a carrier for a recessive trait. If an individual was stillborn, you put the box or the circle and you put a line through it like that. And of course, if the individual was stillborn and the sex was not recorded, you make a rhombus like that and you put a line through it like that. So that's essentially what that means. Spontaneous abortion, you use a little triangle there like that. And then if someone is deceased, then you would put either a line through it, or if you don't want to confuse this with stillbirth, you put a little cross. That's meant to be a cross, not a plus sign. You put a cross next to it, meaning deceased. If you have twins, monozygotic twins, identical twins, you'll do a line and then do a set of lines out like that. And then two circles, identical girls, two squares, identical boys. If they're dizygotic, then you have the line essentially going up to the top line like that. That's dizygotic twins. Dizygotic twins, of course, can be boy, boy, girl, 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 boy. If you have a consanguineous mating, you do two lines between the two circle and square. Mating between relatives would be defined as anything closer, a first cousin or closer, essentially. That is sometimes referred to as a SIB mating, S-I-B, SIB, meaning sibling, SIB mating. Now, let me introduce you to the concept of penetrance. Sometimes you have genetic traits that just don't show up, even though the person genotypically should have the trait. If that occurs, that is called incomplete penetrance. Incomplete penetrance. Incomplete penetrance. If the trait always shows up in someone with that genotype, that is called complete penetrance. Penetrance, based on pedigree analysis, if it's incomplete, you can quantify it. You can get a percentage of the people with that genotype who will show that phenotype. So incomplete penetrance can be quantified as well as it has 50% penetrance or 70% penetrance or 80 or whatever. So let's say, for example, there's a form, for example, of inherited childhood glaucoma. There was a pedigree for this genetic disease that was done in France. And this person went and researched baptismal records, because the baptismal record, the priest would note if the eyes of the child were darkened. If the lenses were darkened, that meant the child had glaucoma, and the child was probably going to be blind. So this researcher went through, and this pedigree was yards long, essentially for families. But it was shown that in that pedigree, there were kids who genetically, this is the dominant trait, who should have been blind, but they weren't. So they had the genotype, but they didn't show the phenotype. That means that that trait didn't always show up when it was expected to show up. That is incomplete penetrance. Incomplete penetrance.
So in their genes, it says they have that.
Yeah.
But they physically don't
Yep.
show.
They have the genotype, but they don't show the phenotype. When I was in graduate school, I worked in the fruit fly with a mutation called jammed. In jammed, the wings of the fly, when the fly wing is made from the imaginal disc, it grows out like this. You get the two sides and the two sides attached together, and you get the two sides of the wing, dorsal ventral. With these, you don't get proper attachment, so you get these blisters and they fill up with liquid. And that liquid, if the fly tries to get out of the pupal case, those blisters burst, and that liquid pastes them to the inside of the pupal case, so they get jammed in the pupal case. That's why they're called jammed. If you took, jammed is a dominant trait. If you raise, say, 100 flies with the jam mutation, all of them are heterozygous for jammed. They should all have blisters on their wings. Well, it turns out of those 100 flies, probably 60 of them, 62 of them or so have blisters. The other 40 don't. So jammed is incompletely penetrant. Excuse me. So here's a pedigree for Huntington's disease. This is a neurological disease. It is a result of a triplet expansion. So in this disease, you have a series of codons, GAG, that code for the amino acid glutamine. You have a stretch of about, 8 of these. When the polymerase gets to this, it can slip. Because it slips, this will have a tendency to expand. If it expands and you get a protein now with large stretches of polyglutamine, that protein will start to, because the polyglutamine tract is a protein-protein interaction domain, it will start to grab proteins in the nucleus and drag them out of solution. And you get these precipitates in the nucleus, and now the neurons will start to die. So this is called a triplet expansion disease. And there's a few others that are triplet expansion diseases. But this disease is dominant. It causes a so-called chorea. Chorea usually involves unwanted movements, uncontrolled movements. If you have someone, for example, hold their hands out, you might see a kind of a writhing of the hand. That's called aceliosis. And so this sort of motion, or you might see a quiver, a tremor, or something like that. So this pedigree, as you can see, these individuals here are affected. This is dominant. This person is unaffected. This person is affected, but because you don't start showing signs of it until middle age, usually at the earliest. This person is homozygous for the wild type allele. And we can see here that 2 of their offspring have that dominant allele. They will have Huntington's disease and so on and so forth. So this is an example of a dominant trait. Now, Huntington's disease, unfortunately, tends to show pretty high penetrance. So these individuals will all tend to develop Huntington's disease. With recessive alleles, you also can have complete and incomplete penetrance as well. Same thing with dominant alleles. So this phenomenon with incomplete penetrance, it's not just the case with dominant alleles, it can also be the case with recessive alleles. Now, don't confuse penetrance with expressivity. Expressivity is different than penetrance. Expressivity refers to the severity of the genetic trait, the degree of the genetic trait. A good example of this, again, comes from my thesis organism, the fruit fly. So, for example, in the wing of the fruit fly, you have these veins in the wing. There's L1, there's L2, there's L3, L4, and then there's L5. Now, there's a mutation called engrailed. If you look at 1,000 engrailed flies, they will all have a gap somewhere here in L4. But for some of them, their L4 looks like that. For some of them, their L4 looks like that. And others, their L4 will look like this. That mutation shows variable expressivity. The severity, the degree of the phenotype varies. So penetrance is whether you see it or don't see it. Expressivity means you see it, but the degree to which you see it, the severity of it, how great it is, how bad it is, or how good it is, I suppose, that varies. So here's an example of a recessive trait. This is albinism. Albinism is recessive. You have 2 individuals here, one of them is heterozygous. You don't know which one. It's known that these 2 individuals are heterozygous. This person is not. So these 2 individuals have albinism, they're albino. These 2 individuals, you can see, have a mating between first cousins. And now this individual has the albino trait. So that's an example of a pedigree, essentially, with a recessive trait. The consanguineous mating there essentially reveals the presence of the 2 heterozygotes. So we talked also about chromosomal mutations. We have alterations in either the growth structure of the chromosomes or the number of chromosomes. So the study of chromosomes, either the number or the structure, that's called cytogenetics. Cytogenetics. There's several ways to get chromosomes for study. You can take some cells, little skin punch biopsy will do it. You can grow them in primary culture, and then you can wait till those cells start to divide robustly, and then you can use a drug called colchicine. Colchicine will arrest the cells at metaphase. The chromosomes are at their largest at metaphase, so they're the most easily seen at metaphase. That's why you use colchicine. Other possibilities, you can use peripheral blood. Red blood cells, of course, don't have chromosomes, but white blood cells do. You can isolate the white blood cells pretty easily from the red blood cells. You can hit them with something that will cause them to divide. Excuse me. Then you can treat those cells with colchicine, lyse the nucleus, and then observe the chromosomes as well. If you want essentially to look at chromosomes from a baby, you could sample the amniotic fluid. That's called amniocentesis. You can't do amniocentesis any earlier than usually 16 weeks of pregnancy. There's not enough amniotic fluid. Before that, it's too risky. Amniocentesis is done with ultrasound guidance. You withdraw a little bit of amniotic fluid, you get the cells from it, you do the cytogenetic studies on the cells, you return the amniotic fluid back to the embryo, the fetus at that stage. You can also, in an earlier embryo, take a look at the chorionic villus. You can do chorionic villus sampling as early as 12 weeks. That involves essentially sampling a small little piece of the growing placenta. That will give you plenty of cells. It grows back just fine. You, of course, can also get cells from bone marrow, culture fibroblasts from a skin biopsy. Lots of options essentially for chromosomes. I must say, because blood is easy, blood is probably the most common. Now, what if you have someone with, say, leukemia? Which of these are you going to use? Bone marrow. That's the source of the white cells. So you want to see the stem cells that are producing the cancerous white cells. So you would do bone marrow. So it's going to depend essentially on what you're looking for. If, say, for example, let's say Maddie wanted to know what her chromosomes look like, because she and her husband want to have kids at some point in time, probably, right? Yeah. We'd probably just take some blood from her. That's easy. That's easy, and she'll just make more blood, and it's not an issue. So when you examine chromosomes, you do something called a karyotype. A karyotype. Now, the way we used to do karyotypes is you get the chromosomes laid out, you take a picture with a camera, and then literally you blow the picture up, and then you cut it out with scissors, and you'd lie them all by size, and essentially just get it like that, and then examine the chromosomes that way. Today, you don't have to do that because computers will put the chromosomes on there, then you hit a button that says sort by size, and the computer goes and does all that for you. Something that took hours in the past, now you can do with a push of a button, and it'll do it within 120 seconds. So yes, I hate them all because I wasted my time. No. So karyotyping has become much easier. Now, you have to stain the chromosomes in order to be able to see them. What do you stain them with? What do you want to look at? If you want to look at gross chromosomal structure, normally what you do is you stain them with a Giemsa stain. Bless you. Giemsa stains are easy and they're cheap. And they will give you a rough idea of the gross chromosomal structure. You'll get banding patterns. Those banding patterns will be specific for each particular chromosome. And based on size and banding pattern, a good cytogeneticist can look at it, a spread in the microscope, and say, oh, there's 1, there's 22, there's 16, there's, and what have you. If you do this enough, you can, I'm not that good, but with Drosophila chromosomes, I was pretty good at it. But of course, Drosophila only has 4 chromosomes, so I suppose that's cheating. But anyway, they're numbered from 1 to 22. It says 22 is the shortest. That's actually wrong. 21 is the shortest. 22 is a little bit bigger than 21. So 21 should actually be 22, but historically it's known as 21. So 21 is actually the shortest. And then the sex chromosomes, the Y chromosome is very small. It's pretty easy to spot, frankly, because it's so small. And then the X chromosome has a morphology that's pretty easy to see. Even a novice like me can detect it. If you want to know the location of a particular gene on a chromosome, then you would do this thing called fluorescence in situ hybridization, FISH. And that is done under fluorescence. So here's an example of these chromosomes. That's a little better. Can you see the banding patterns here? Can you see those? Okay, so the ones in front can see it. I don't know the ones in back can see it, but there's these spots that are well stained and then other spots that aren't. See the open spot there? The X looks like a sausage that you rolled on a barbecue. It looks like brats you rolled on a barbecue. That's how you can tell the X. It's pretty easy to spot. The first chromosome, do you see that? See that pattern like that? That's very characteristic of the first. Plus the first is really big. Yeah, just patterns like that you get to see. So these are stained with the Giemsa stain. And they're photographed with a microscope. And again, you don't have to sit there and cut them out. You can just push the button in like that. And some of you may get pictures like this if you order, or if your boss, the provider, orders tests like that. You may see stuff like that. You may see things like 321s, or you may see chromosomes where one chromosome is a little shorter than the other. That usually indicates you have a piece of the chromosome that's missing. You have a deletion, a deficiency. Or it might mean the other chromosome has a duplication or something like that. That's called chromosome banding. And then FISH is for viewing individual genes. Let's say you have reason to believe that somebody has a piece of a chromosome missing, or there's such a mutation that they don't make the gene. Well, you can then test that essentially. Does this chromosome essentially have this particular gene? And FISH technology has gotten to the point now where you can screen one particular experiment, you can screen for multiple genes if you want to look at multiple genes. So FISH has become very, very effective. So to do FISH, you have a piece of DNA that can hybridize to that gene. That piece of DNA is called a probe. When you make that DNA, you make it with nucleotides that have dyes attached to them. So when these probes hybridize to the chromosome, they will fluoresce. If they don't hybridize, they're not going to fluoresce. And so you label it with fluorescent dyes. The chromosomes, then you take alkali, you just put some alkali on them that denatures, takes the double strands, unwinds them. You then give it this probe that you made. It'll hybridize to this probe. You use a solution that will allow it to hybridize properly. And then you view it essentially under a fluorescent microscope situation. And it tells you if that gene essentially is there where it should be or something like that. So cytogenetic analysis can tell you essentially if you have a particular chromosomal aberration, either number or structure. You can also use it if you have some sort of developmental issue. Now, developmental issues may be due to mutations in single genes that you can't detect with just cytogenetic analysis. That may require more detailed genetic analysis. If you have other sorts of things going on, if someone has spontaneous abortions of unknown cause that keep repeating, then you might want to get the mother and the father subjected to cytogenetic analysis. Malignant tissue or pre-malignant tissue, cytogenetic analysis can be incredibly informative. Parents of a patient who has some sort of chromosomal translocation, other types of stuff, prenatal diagnoses, all of these essentially are things that for which you would use cytogenetic analysis. What about testing for individual genes? It gets a little trickier because some tests for certain genes are commercially available. Insurance will pay for them. These tests are not cheap usually. So say, for example, let's pick on Erin back there. Let's say Erin in her background has Ashkenazi Jew in her background. So she's from Eastern Europe, Jewish population, and she meets the man of her dreams. They get married, they want to have kids. Now, they meet with their OB. The OB says, tell me about your background. And Erin says, I'm descended from Ashkenazi Jews. And the OB says, Tay-Sachs. Tay-Sachs frequencies are relatively high. In Ashkenazi Jewish population, you might carry the Tay-Sachs allele. You should probably be tested for it. And Erin says, okay, pulls up her arm, says, take it. I've got good veins, see? That test is paid for by insurance. That test is easy to do. It's been done so many times, it's relatively inexpensive, and insurance will pay for it. Let's say she wants now to be tested for some very rare genetic disease. A test that has to be sent to a university lab because there's no commercial test available for it now. Insurance will not pay for that. And that test, I will lay you money, is going to be ridiculously expensive. Also, you can't test for everything. You can't. It's impractical. So, word of the wise about these genetic tests, use them when necessary. Use them when necessary, because sometimes they may tell you very useful information. Other times they may not. And if they're not going to give you useful information that provides you with good answers, that will give you a good therapeutic differential, forget it. Forget it. It's a bunch of stress and expense and worry that you don't need and your patient doesn't need, right? So these are to be used with a degree of judiciousness. All right, and the big thing is there a family history of it? If there's not a family history of it, then guess what? Your patient's a worry wart. Should you indulge them? No, no. Don't indulge destructive behavior. Oh, but it'll give them peace of mind. No, it won't. They'll just come up with another genetic disease that they'll think their kid might have. No, do not indulge destructive anxiety. Use these only when they're going to give you useful information. Okay, we'll finish this on Friday. Don't forget, we have a test on Monday. And because we have a test on Friday, we will not have Monday will not have a quiz on Friday. Okay? Now I know you're all just dying for a quiz, right? Right, okay. No quiz on Friday. How you doing? Good. No.